John Vane was awarded the Nobel Prize in 1982 for his work on really the mechanism of action of non-steroidal anti-inflammatory drugs, aspirin and indomethacin, and also developed new treatments for hypertension through ACE inhibitors, and so a very important contribution. The John Vane Medal is awarded in different subjects in different years, and this year it was in the area of drug metabolism and pharmacogenetics, and so since it is an area that I've worked in effectively since the early 80s, it was absolutely wonderful to be able to describe a whole body of work over a prolonged period, and that's what this medal lecture has allowed me to do. It's a great honour to be chosen, and particularly since I'd met John Vane on one occasion when he opened the pharmacology department in Oxford officially in 1991. As well as being an honour, it's also been really a lovely thing to do because I've been able to think about all the people who've worked with me, and also why I did the various experimental studies that I did, and particularly to be able to reflect on the importance of undergraduate project students and master's students, as well as the PhD students and postdocs and collaborators who've been part of the team. First of all, we started working on N-acetyltransferase because it was the most important polymorphism in understanding a condition called hydralazine-induced lupus, and that is an adverse side effect. And this was in the early 80s, when the whole genome approach to understanding contributions to multifactorial diseases hadn't been established at all, and so we took a candidate gene approach, and from previous immunological work that I'd done, it was clear that hydralazine lupus was a good way of looking at gene-environment interactions. Who was it who got the disease compared with who didn't get the adverse side effect? And it became clear at that stage that the most important polymorphism was the ability to metabolize hydralazine, and therefore I started work on that, and it's kept me amused for almost 30 years. It was a very wide-ranging talk, and I had spoken about the enzyme that I've worked on most of my life, Arylamine N-acetyltransferases, or NAT for short, is found primarily as a drug metabolizing enzyme in humans and other mammals, but our own work has identified it as a target for anti-tubercular therapy in Mycobacterium tuberculosis. The interest in the enzyme in Mycobacterium tuberculosis is particularly because in humans it actually metabolizes an anti-tubercular drug, and that anti-tubercular drug needs to be activated in Mycobacterium tuberculosis, but if NAT comes along, then that would stop it being activated. So we'd had thought initially that the enzyme in M. tuberculosis could contribute to isonases resistance in the organism, but actually what we found was much more exciting than that. What we found was that actually it did contribute a little to isonide resistance, but the important point was that actually it itself is essential. The gene itself in Mycobacterium tuberculosis is essential for survival of MTB in macrophage, and my own work has moved on from there, looking at other genes in the same gene cluster. The whole gene cluster is essential for survival of M. tuberculosis inside macrophage. From these studies, work that we did with a spin-out drug company from Oxford, where I was working at the time, was related to that has now been taken up for license to develop as anti-tubercular agents, and that's been very satisfying.